Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine these incredible cutting-edge medical treatments. We're talking about using your own immune cells, engineered to fight cancer, or delivering tiny packages of therapeutic genes right where they're needed.
Really powerful stuff. Precision technology.
Exactly. But here's the catch. Once those living cells or those gene packages are actually inside your body, seeing exactly where they go and what they're doing, that's been incredibly difficult.
Yeah, it's like, well, the analogy in the outline was good, sending specialized troops behind enemy lines, but having no tracking, no comms.
Right. You just hope they get there and do the job.
Yeah.
You can't really watch the mission unfold.
And that lack of real-time visibility, that's a huge bottleneck. It really is, for developing these therapies, for using them safely.
You've got these dynamic, living things working inside a super complex system, the human body.
And clinicians just don't have a standard way, a clinically useful way to actually observe them over time.
Kind of a black box situation, isn't it?
Very much so.
But what if you could look inside that black box? That's the really exciting part of the new research we're diving into today. This work suggests a new technique, one that lets researchers track those modified immune cells, like the ones in ESI-R T-cell therapy. That's chimeric antigen receptor T-cell therapy.
All right.
And also monitor gene therapies. And they do it using a pretty standard medical imaging tool, PT scans, positron emission tomography.
Yeah, and this deep dive, it's based on a Health News article covering the research, but also, importantly, the actual scientific paper, the one that gets into the real nuts and bolts from the researchers at the Technical University of Munich.
Okay, so our mission today, let's unpack this. We want to understand what this new tracking method really involves. What's the clever idea behind it? What did the researchers actually find when they tested it out? And maybe the biggest question, why does this even matter? Why is seeing these therapies inside the body such a big deal for their future? And maybe for you, listening down the line.
Okay, so let's start with that challenge. You mentioned CAR T-cell therapy. It's really changing the game for some diseases, right? Especially cancers that haven't responded to other treatments.
Absolutely.
The basic idea is you take immune cells from a patient, tweak them in the lab so they learn to spot and attack specific targets on disease cells like cancer cells.
And then you put these sort of souped up cells back into the patient.
Exactly, infuse them back in.
And the hope is they multiply, hunt down the disease wherever it is, and, well, get rid of it. Sounds great on paper.
It does. But here's that problem again. Once those modified cells are back inside you swimming around, there's no routine clinical way to see precisely what they're up to.
That's the core issue, isn't it?
It is. I mean, think about the questions you'd have if you're the doctor or the patient. Do the cells actually get to the tumor? Or the right organ?
Yeah, are they multiplying like they should?
Or are they just fizzling out?
Or, crucially, are they staying put? Or are they maybe wandering off and attacking healthy tissue? That can cause serious side effects. And without seeing them directly over time, answering those questions in a real patient setting is just really hard.
Incredibly difficult. And the ways we've tried to track cells before, they just don't cut it for this kind of thing, Whole body, long-term monitoring.
Right, like putting a label on them before infusion.
Yeah, you can do that. But every time a cell divides, that label gets diluted, split between the two daughter cells.
And these therapies, like made CAR T cells, they need to divide to work. That's the whole point, often.
Exactly. So that initial label just fades out pretty quickly. It doesn't last.
Okay, what about optical imaging, using fluorescence?
Fantastic in a lab dish. Or, maybe just under the skin. But light just doesn't travel deep into the body.
So you can't see what's happening in the liver or the heart or deep in the bone marrow.
Nope, not going to work, which is where PET imaging comes in. It can see deep inside the body. It's used in hospitals everywhere.
There's always a but.
But you need a way to tag the specific cells you want to see. You need a compatible reporter system, something on the cell that a PET tracer, that's the special radioactive molecule, can latch onto.
Making the cell light up on the scan.
Right. And finding the ideal reporter system, one that's specific, safe, works well in vivo inside a living body. That's been a real challenge.
Okay, so challenge established. Powerful therapies, need to see them work. Current methods lacking. PE needs the right tag. Now, the solution from Munich, this sounds like where the clever engineering comes in.
It really is the core idea. They modify the therapeutic cells, the immune cells, or even the gene therapy delivery systems to carry a second, totally artificial protein receptor on their surface. Or for gene therapy, to make the cells express this protein after the gene transfer.
A second artificial receptor, okay. Where did that come from?
Yeah, this is the key bit. It's based on a family of natural proteins called anticalins.
Anticalins.
Right. Naturally occurring proteins that bind things. But Professor Arne Skara, one of the researchers involved, he's been a pioneer since way back in the 90s in engineering these anticalins in the lab, basically reprogramming them.
To do what?
To bind really specific targets, often artificial molecules they wouldn't normally interact with.
Got it. So they took that expertise.
And they designed an anticalin to bind to a very specific small molecule known by the abbreviation DTPA.
Okay.
Then they adapted this engineered anticalin to create an artificial protein that sits right on the cell surface. It acts like a receptor, and they just call it the DTPA receptor.
Simple enough. So the therapeutics, say, the CRT cell, now has this extra artificial DTPA receptor stuck on its outside.
Exactly. Now, to see it with PET, you need that compatible tracking agent, the radioligand.
The choice for molecule.
Right. So they developed a special radioactive contrast agent. It's designed specifically to bind only to that artificial DTPA receptor. And importantly, it seems to be non-toxic.
Okay, so let's walk through it. You got the cell with its unique DTPA receptor flag. You inject this special radioligand. What happens?
So this radioligand, they use a version labeled with fluorine-18. That's a really common radioactive tag for PET stands. So fluorine-18 DTPA. You inject it, it goes around the body. And this is critical, it's designed to only bind very specifically to that artificial DTPA receptor, which is only found on the engineered therapeutic cells you put in.
Okay. So it's like they built a custom-made lock, the DTPA receptor, onto the therapy cells.
Yeah.
And this fluorine-18 DTPA is the perfectly matched key, floating around the body, only looking for that specific lock.
That's a perfect analogy. And because it binds so specifically, the fluorine-18 radioactivity builds up wherever those engineered cells are located.
And fluorine-18 gives off positrons,
which the PET scanner detects.
Yeah.
So this accumulation of the radioligand makes those cells light up on the scan. It lets researchers see where they are and how many are there, non-invasively.
That is really elegant. A highly specific engineered reporter system that plugs right into existing PT scanners.
Yeah, very smart.
So what did they actually find when they tested it? The paper mentions a lot of mouse studies.
Right. They tested it extensively. The results were pretty compelling, especially looking at the CAR T cells first, since that's such a hot area right now.
Did the cells go where they were supposed to? Did they reach the disease?
They did. The PE scans using that fluorine-18 DTPA clearly showed the modified CAR T cells migrating to the diseased tissues in the mouse models.
Okay.
And importantly, they saw them multiplying there too, which is exactly what you need for CAR T cells to work effectively.
And could they watch this happen over time?
Yes, they could track this expansion, this movement over quite a while. The paper mentions up to 30 days.
30 days. That's a whole month of tracking the same cells. That blows direct labeling out of the water.
Absolutely. Far beyond what you could do with just sticking a label on at the start.
Was the signal really specific? I mean, did it only light up the engineered cells, or was there background noise?
The specificity looked really good, very high. The PET signal was strong only where those cells with the artificial DTPA receptor were.
And the control group. Mice that got TRT cells without the artificial receptor.
Critically, those control mice showed no significant signal. That really confirms the signal was coming only from the cells carrying their engineered tag. It wasn't just sticking randomly.
Okay, so it gives a clear picture. But could they get more than just a visual? Could they actually estimate how many cells were in a certain spot?
Yes, and this is a really crucial finding from the paper. A major strength. They showed that the brightness, the intensity of the PET signal in a tissue linearly correlated with the actual number of CAR T cells present there.
Linearly correlated, meaning more signal equals more cells in a predictable way.
Exactly. They confirm this using other lab methods too, like quantitative PCR. It means the technique gives you quantitative data, real numbers. It's not just seeing if the cells are there, but getting an estimate of how many are actually on the job fighting the disease in real time.
That is huge. For research, definitely. But imagine for clinical use eventually getting an actual count. How sensitive was it? Could they pick up small numbers of cells?
Pretty sensitive, yeah. In the living mouse models, they reported detecting as few as around 1,200 CRT cells in bone lesions.
Inside the bone, that's impressive.
It is. And in lab tests, even fewer, maybe 500 cells. If they injected them just under the skin, they could clearly see about 8,000. That sensitivity, especially deep inside the body, is significant.
Okay, and you mentioned they didn't just stop at CRT cells. They also applied this to gene therapy.
They did. They adapted the same reporter idea to track a common gene therapy delivery vehicle, adeno-associated virus type 9, or AAV9.
A virus used to shuttle genes into cells.
Right. They engineered these viruses so that after they delivered their gene payload, the cells that received the gene would then start producing that same artificial DTPA receptor protein.
Ah, so the receptor acts as a marker for successful gene delivery.
Exactly.
And what did the PT scans show there?
Similar story. They could track where the gene transfer actually happened in the body after they injected the virus. And again, crucially, the PT imaging allowed for a quantitative analysis.
Meaning they could estimate how much gene transfer occurred in different organs.
Precisely. They looked at organs where this virus is known to go: liver, heart, muscles, adrenal glands, even brown fat. The PT signal strength lined up nicely with the dose of the virus they injected.
And did it match up with later checks?
Yeah, it correlated well with histological analysis afterwards, which confirmed the gene expression in those tissues, so it reinforces that quantitative aspect for tracking gene delivery too.
That's really consistent across both cell and gene therapy applications. What about the radioligand itself, the fluorine-18 DTPA? Was it safe in these studies? Did it hang around too long or go where it shouldn't?
The studies indicated it cleared out pretty quickly, mostly filtered by the kidneys and excreted in urine. And it showed very low non-specific binding elsewhere in the body. It really seemed to stick mainly where that artificial receptor was.
Did it seem to mess with the cell's function?
Not according to their lab tests. It didn't appear to interfere with the function or activity of the therapeutic cells themselves.
Okay, let's pull back for the big picture then. We have this method-specific, quantitative, works deep in the body for both cells and gene delivery. Why is this such a big deal? What does it really enable?
Well, it's a massive step towards something the field has desperately needed: non-invasive, whole-body monitoring of these incredibly complex living medicines.
Instead of biopsies or just guessing based on indirect science.
Exactly. Right now, finding out where cells or gene vectors have gone often means invasive procedures or relying on blood markers that don't tell you where things are happening. This offers a way to see the whole picture, the biodistribution, how things behave over time, without cutting into the patient or the research animal.
So it gives researchers, and maybe one day doctors, just unprecedented insight.
Absolutely unprecedented insight into where the therapy goes, the biodistribution, how long it lasts and stays active, its persistence, and what it's actually doing, its behavior. Is it growing? Is it hitting the right targets? Is it going off target? All seen non-invasively in a living system.
And that kind of insight, it sounds like it could directly impact safety.
It really could if you can see early on that cells are maybe accumulating in a healthy organ where they shouldn't be, or multiplying way too fast.
You get an early warning.
Precisely. A PET scan could provide that warning, potentially allowing doctors to intervene before serious side effects develop. That could make these powerful therapies significantly safer.
And what about just developing new therapies? Does this help?
Hugely. Being able to track quantitatively and non-invasively right from the early animal studies through potential clinical trials, that could massively speed up development and evaluation. Researchers can get objective data comparing different versions of cells, different doses, different ways of delivering them, see directly how they behave in the body. That speeds up the whole optimization process, finding what works best faster.
Sounds like it could streamline the preclinical work too. Maybe use fewer animals.
That's another potential benefit, yes. It's incredibly valuable for the basic research, just understanding how these things work inside the body. And yes, potentially reducing the number of animals needed because you can monitor the same animal repeatedly over time instead of needing separate groups for each time point.
It really feels like a foundational tool they've built. There's that quote from Professor Wolfgang Weber, one of the lead researchers.
Yeah, we believe that we have created a valuable tool that can make such therapies safer by providing better insight into what happens inside the body.
That sums it up pretty well, doesn't it?
It really does. Moving from that black box scenario to something much more transparent, much more observable.
Of course, we need the reality check. This is research. It's exciting, it's promising. But where does it stand now? What needs to happen before you or I might see this used in a clinic?
That's the crucial caveat. Yes, the mouse studies, the lab work, very encouraging. But it's still early days for human application.
So, not ready for prime time just yet.
Not yet. The next big step, the absolutely essential step, is rigorous safety and efficacy studies in human clinical trials. That's non-negotiable.
And that takes time and careful work.
Exactly. Development towards clinical use is ongoing, but it involves navigating complex regulations, proving safety, proving reliability in people.
Did the paper mention any specific hurdles they anticipate for human use? Technical challenges.
They did touch on a couple of important points to watch. One is always the question of immunogenicity. Could the human immune system react against that artificial DTPA receptor protein?
Because it's technically a foreign protein.
Right. Introducing any engineered protein raises that possibility. They said the preclinical results in mice were encouraging on this front, and there's some positive history with other engineered anticalins, but it needs very careful evaluation in humans.
Makes sense. Anything else?
The other point was more technical, achieving really high specific activity when they make the fluorine-18 DTPA radioligand. Basically, getting enough radioactive fluorine-18 onto the DTPA molecule efficiently and consistently. That's a common challenge for many PT tracers, but something they need to optimize for reliable, large-scale clinical use.
Okay, so exciting progress, a clear path forward, but definitely still a path that needs careful navigation.
Exactly. A significant leap in what's possible, opening doors, but not quite at the destination yet.
So let's wrap this deep dive up. We've got these researchers from the Technical University of Munich who developed a really unique way to use PT scans to see engineered cells and gene therapies inside the body.
Yeah, with high specificity, seeing only the target cells and even getting quantitative data, an idea of how many cells are there.
And they did this by giving the therapeutic cells or vectors an artificial protein flag, that DTPA receptor, which is then specifically found by a radioactive tracer, fluorine-18 DTPA, making the cells visible on a standard PT scanner.
And this tool, it really holds huge potential. It could transform how we understand, monitor, and maybe even control these powerful living medicines, the ultimate goal being to make them safer, more effective as they become more common.
It really does make you think, doesn't it? As these personalized cell and gene therapies advance, as they become more part of medicine, imagine being able to literally see those living treatments working inside your own body.
Yeah.
How does that change how you understand your health, your treatment? What possibilities open up when you have that kind of precise, real-time monitoring, truly tailored medicine, maybe adjusting treatments based on exactly what's seen happening inside you?
It's a fascinating future to contemplate, and this research brings it a little closer into view.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.